So I would like to thank the organizers for inviting me to give this introductory talk. And
That's forward there. Back.
by way of disclosure, I was not on the organizing committee when I was invited, but I was asked to join the organizing committee. So like Francis, I was somewhat confused as to how to present a talk with such a diverse audience. Hearing everyone's introduction makes me think the audience is even more diverse, but I think I've included something for everyone in my talk, from the very basics on kidney function to intricacies of type 4 collagen and the glomerular basement membrane. So here's an outline of my talk. I'll begin by talking about briefly kidney structure and function, move on to the glomerulus and the glomerular filtration, showing you in action how it works, and then talk about in detail the glomerular basement membrane, defects in Alport syndrome, the molecular architecture that we've been viewing it using super resolution microscopy, and finally, evidence that the GBM is a plastic, malleable, changeable structure. And I will, at some point, need the lights down, so I hope that will be possible, because I have some rather dim fluorescence to show. But to begin with, the kidney is composed of, at least the human kidney, of about 1 million different nephrons, which are individual filtration units. One of them is diagrammed here. It begins with the glomerulus. The glomerulus is highly vascularized, and the filtrate that's formed in the glomerulus flows down the tubular portion of the nephron, where it is altered by the cells that line the tubule, such that it ends up as urine, which then enters the collecting system, eventually makes its way to the ureter, and then to the bladder, where it's stored until it can be properly eliminated. So a little bit of renal physiology. Human kidneys generate an enormous primary filtrate, about 180 liters per day, from about 900 liters of blood that pass through them each day. But urine output is only 1 to 2 liters per day, and that's because most of the water that's in the primary filtrate is reabsorbed by the cells that line the tubular segments of the nephron. An important feature of the glomerulus is that very little plasma protein passes through that filter. So if there is elevated protein, especially albumin or any blood in the urine, this indicates that there is some sort of kidney disease or injury. So here is a schematic diagram of the glomerulus that I show in just about any kidney talk that I give. It's basically a cross-section looking at the glomerulus, and so you can see the glomerular capillaries are in cross-section here, so blood flows through here. And then the urinary space, also called Bowman space, is this part of the kidney here, which contains the primary filtrate. So the kidney has 3 major cell types. The glomerulus has 3 major cell types. Like all blood vessels, the glomerular capillaries are lined by endothelial cells, but the glomerular endothelial cells are specialized because they have little windows or fenestrations that allow the plasma flowing through the bloodstream to come into close contact with the glomerular basement membrane, which is this ribbon-like structure here. On the opposite side of the basement membrane from the endothelial cell are cells called podocytes, which are the cells that secrete the collagen 4 alpha 3/4/5 network that's affected in Alport syndrome. And the endothelial cell also secretes some components of the basement membrane, but not the alpha 3/4/5 collagen chains. The third cell type is the mesangial cell, which are smooth muscle-like cells that bind to the bases of the capillary loops and maintain these capillary loop structures. And by contracting, mesangial cells can actually change the diameter of the glomerular capillaries and change the hydrostatic pressure of the plasma that's flowing through the capillaries, and therefore influence the rate of filtration across the capillary wall. So here's where I think I need the lights down a bit, and hopefully I can get this movie to play. Looks like it will work. So this is a movie made by Bruce Molitoris at the University of Indiana in Indianapolis, looking at the glomerular filtration in a live rat kidney. And the rat has been injected intravenously with a yellow substance that is large and with a red substance that is small. And you can see that, for the most part, all of the yellow substances stay in these glomerular capillaries. These are other capillaries outside the glomerulus. This is the glomerulus here. Whereas the red substance flows across the filtration barrier into Bowman space and down the tubule. So basically, this is demonstrating glomerular perm selectivity, whereby large molecules are retained, small molecules are filtered, and eventually will end up in the primary urine. So what about this filtration barrier that is affected in Alport syndrome and in nephrotic syndrome, which I know a lot of you also have indicated that you are interested in? So this is looking at this podocyte that lies on top of the glomerular basement membrane by scanning electron microscopy, showing many processes that these podocytes extend that interdigitate with foot processes from adjacent podocytes. And that covers the outer surface of the basement membrane. If you look in cross-section through this capillary wall, you can now see these podocyte foot processes individually sitting on the glomerular basement membrane, which is that structure there, and then the fenestrated endothelial cell that lines the inner part of the capillaries. So this is the entire filtration barrier, basically, between the blood and the urinary space, and this is what is affected in Alport syndrome, this glomerular basement membrane, whereby rather than having this nice ribbon-like structure with the individual foot processes on top, the glomerular basement membrane becomes split and thickened, and eventually, though not at first, the podocyte responds by retracting those processes, losing the spaces between the processes through which filtrate must flow. So this glomerulus would be unable to filter as well as this glomerulus because there isn't enough space for the filtrate to flow across. In addition, the defect in the basement membrane can have numerous alterations on the glomerular barrier such that molecules such as albumin and other proteins would more readily cross into the urine. And that's how protein shows up in the urine. A student in my lab, Jung-Hee Seo, was interested in looking at the basement membrane in Alport syndrome versus normal. Because we don't have access to human Alport kidneys, we used our mouse model of Alport syndrome, which is a collagen 4 alpha 3 autosomal recessive version of the disease. And what she found is that in the normal glomerulus, this is now taking away all the cells and isolating the glomeruli. In the normal glomerulus, the basement membrane looks like kind of a bow that you would put on a present for Christmas, a ribbon-like structure. But in Alport syndrome, the basement membrane is much different. It has blebs, and I'll show it to you at higher power here. You can see it's very blebbed, and this probably represents those outpocketings that are typical transmission electron microscopy diagnostics that you're seeing now in a more 3D view. And so you can imagine that the podocyte, which normally sits on a flat basement membrane of a certain composition, in Alport syndrome is sitting on this very abnormal basement membrane of abnormal composition, and that can contribute to the eventual podocyte injury and other effects of Alport syndrome. So this is a fluorescence micrograph that I think I made in 1994 of a rat kidney using antibodies that I had made to one of the collagen alpha 3, 4, or 5 chains, I forget which, but that's what's shown in green. And in red is an antibody to the alpha 1, 2 type 4 collagen trimer. And you can see that the green, where the alpha 3, 4, 5 chains are in these peripheral capillary loops of these individual capillaries within the glomerulus. For those of you on that side, here's one here. Whereas the red is primarily in the mesangial matrix, though there also is some overlap between the red and the green at the junctions where the mesangial cell binds the glomerular basement membrane. So the alpha 3, 4, 5, as Francis told you, is the network that's present in the glomerular basement membrane. But in Alport syndrome, at least in the Alport mice that we have, but not in all patients, this network is totally missing. These chains cannot be detected in the glomerular basement membrane or anywhere else, and that's because in the absence of alpha 3, this network simply cannot form. Instead, there's compensation by the alpha 1 and alpha 2 type 4 collagen network such that these chains appear more apparent in the glomerular basement membrane, but of course they don't function properly. Whatever function collagen alpha 3, 4, 5 has, alpha 1 and 2 cannot fully compensate, but at least they compensate to allow a basement membrane that functions for years to be able to form. So I wanted to point out in this different drawing of a nephron. So here's the vascularized glomerulus, that when these capillaries and the cells that line them become injured in Alport syndrome, those cells begin to make extra matrix, and the glomerulus becomes sclerosed or hardened, which prevents blood from fully flowing through them, such that filtration in the glomerulus won't happen at the rate that it needs to maintain normal kidney function. But in addition, these capillaries actually lead to the intertubular capillaries. So if blood flow is blocked here, that leads to blood flow being blocked in other parts of the kidney that surround the tubules, which can lead to injury beyond the glomerulus, which is the site where the alpha 3/4/5 network needs to be deposited. So in Alport syndrome, these GBM structural defects eventually lead to increased expression of other matrix proteins and proteases. Work from Dominic Cosgrove that he might talk about tomorrow might address that. There's glomerular scarring and infiltration of inflammatory cells due to this injury, both in the glomerulus as well as in the interstitium, which is the part of the kidney between the tubules. Both become invaded by inflammatory cells. There's obstruction of the capillaries and reduced renal blood flow and glomerular filtration rate. So this is a typical end-stage kidney from one of our Alport mice, where this mouse was near death. Its kidney is shrunken and yellowed compared to its normal littermate kidney, which is still red because so much blood is flowing through it the way it should be. So alpha 3/4/5 is not the only glomerular basement membrane protein. Laminin 521 is another major one. There are two proteins, different versions of a linker protein called nitogen, and then agrin, a heparan sulfate proteoglycan. We should hear from Rachel Lennon tomorrow, I believe, that there are many more proteins found in the glomerular basement membrane. Look forward to her talk. And so if one, based on biochemistry done by a number of different labs over the years, one can think of a way that these four major proteins can assemble within the extracellular matrix to make a basement membrane. This is a version created in cartoon form by Peter Yurchenko. But is this really an organized extracellular matrix, or is it a mishmash of all these proteins together? And secondly, can understanding exactly how these proteins are organized in the GBM impact our understanding of Alport syndrome? So this is a question that my lab and collaborators at Washington University, Andre Shah and Adish Dani, addressed using a new super resolution microscopy method to look at the location of individual proteins within the glomerular basement membrane. And we're able to do this at the epitope level, so using antibodies specific for known epitopes present on these proteins, for example, the agrin N terminus, the agrin C terminus, the laminin alpha 5 LG domain, the laminin alpha 5 LEB L4B domain, the collagen 4 NC1 domain, alpha 3/4/5 NC1 domain, or an N-terminal domain. We were able to map all of these epitopes within the glomerular basement membrane and see how these proteins are actually arranged. And these data are published, so I'll just summarize and show you the highlights relevant to Alport syndrome. One surprising thing we found was that in both humans and mice, we really didn't have much trouble detecting the alpha 1, alpha 2 network, but it was only found on the endothelial side. And this is consistent with data published in the literature many years ago. I believe Cliff Kashtan may have been involved in that study, but I know Minneapolis was involved in that study. And we could double stain with an antibody to the extracellular domain of a receptor that binds matrix proteins called integrin beta 1. And you can see the integrin beta 1, this is now the podocyte side up, is present. It's present also on the endothelial cells, and it's graphed here. So the blue is integrin beta 1. So you're seeing a endothelial peak and a podocyte peak. Most of the alpha 1/2 network is directly adjacent to the endothelial integrin beta 1. Now, in contrary, the alpha 3/4/5 network, and this is human kidney now, is present in the very middle of the glomerular basement membrane at a site that seems to be too far away from the integrin beta 1 that's present on either podocytes or endothelial cells, such that it's not likely that cell surface receptors, either on podocytes or endothelial cells, is able to bind the Alport collagen network. And that is summarized in these statements here. So now I'll go on. And so by doing this with multiple antibodies to different matrix proteins, we were able to basically generate a map of where these protein epitopes are localized within the glomerular basement membrane. And the take-home messages are that the integrin beta-1 here is quite close to the C-terminus of agrin and the C-terminus of laminin alpha-5. But the integrin is not really near the N-terminus of laminins or the N or C-terminus of type 4 collagen alpha-3, 4, 5. But as I stated, the alpha-1, 2 is closer to the endothelial cell. And so based on, and this is the graphical representation, should be able to bind. We then used our Alport mouse model. Unfortunately, we couldn't get our hands on either human or dog Alport kidney to do this in those species. But what we found was that, as expected, there was an expansion of the alpha-1, alpha-2 type 4 collagen network from specifically being on the endothelial side towards being more increased on the podocyte side, which is this side here, such that podocytes should be able to come into contact with the alpha-1, 2, 4 network. And so it seems that this network shifts to the podocyte side and should be able to interact with the beta-1 integrin on podocytes. And so this suggests the existence of a collagen 4 signal to podocytes in Alport syndrome that is not normally present. Now this is kind of a provocative hypothesis that we don't really have proof for yet, but there are receptors on podocytes that have been studied by others, such as integrins, DDR1 that Oliver Gross has studied, that could suddenly see type 4 collagen, whereas the podocytes would not normally see that signal. And so that could lead to downstream effects that eventually lead to glomerular sclerosis and Alport syndrome. And showing that here, so this podocyte is now seeing a type 4 collagen that we think the normal podocyte doesn't see. It's only seeing laminin and agrin in the normal state, but is also seeing type 4 collagen in Alport syndrome. So moving down, as most of you know, there were some provocative papers in the literature in the mid-2000s about the possibility of using bone marrow stem cells and other cells to introduce into Alport patients or mice and get those cells to basically infiltrate the glomerulus, become podocytes or fuse with existing podocytes to then restore expression of the type 4 collagen alpha-3, 4, 5 network. I wasn't totally convinced by a lot of these data, and I know a lot of you, or some of you in the room, also were not convinced. This is one example of data I didn't find convincing. Here's a knockout mouse lacking alpha-3, 4, 5. This is a normal mouse, which has alpha-3, 4, 5 in the glomerular basement membrane. And this is after an Alport mouse after a transfusion of cells, purportedly now having the alpha-3, 4, 5 network in the GBM. So one question to me was, well, once you have an Alport GBM, can you really change it, normalize it, by putting back in the missing alpha-3, 4, 5 type 4 collagen? These investigators used an adult mouse, which would have had a GBM that's abnormal and functioning, but could it really be modified to the extent that it could restore normal kidney function or normal structure? And so I decided that we should take a step back and use the power of mouse genetics to really answer this question. Were gene therapy possible in humans if we could just snap our fingers and fix the mutation in alpha-5 in some of you, or put back in the alpha-5 gene? Would that impact your kidney function? Would that rescue your basement membrane? So we used mice to do this using an inducible system. We can do lots of things in mice that we can't yet do in humans. But we basically use a system in which we could induce expression of the alpha-3 chain in our alpha-3 knockout mice at any time point using an antibiotic called doxycycline. And so if we do this on the Alport background from before birth or at birth, we basically have a rescued mouse that has a normal glomerular basement membrane because we've basically rescued the gene defect by re-expressing the missing protein. But the real question here is, can we restore the missing alpha-3, 4, 5 after the GBM is mature and functioning. And so we basically, and the way we did this experiment was to only induce expression in podocytes based on work by Dale Abrahamson in Kansas City, who showed that only podocytes make the alpha-3-4-5 network. And so what we found was that if we induce at 3 weeks of age, which is a point which might be mid to late adolescence in a mouse, or represent mid to late adolescence in a human, though that's a debatable point, we could get significant rescue. First, I'll show you that if we induce from the day of birth, we can now replace the missing alpha-3-4-5 network. And this is using an antibody from Bogdan Borza in Nashville that only recognizes the hexamer, the formed hexamer whereby the alpha-3-4-5 network is properly assembled. So this antibody doesn't stay in the alpha-3 knockout because there is no alpha-3-4-5. It does stay in the wild type. There's alpha-3-4-5 in the glomerular basement membrane as well as in tubular basement membranes. But we've only induced expression in podocytes, and so you only see the glomerular basement membrane positive. If we induce as late as 3 weeks of age, after 20 weeks, this is what we see. We see that basically the network is there, even though the basement membrane began as an abnormal alpha-1, alpha-2 only basement membrane. But can we, and this is work that we published recently in the Journal of American Society of Nephrology. We've also done additional studies where we induced at later and later ages to see how applicable might this be to adult humans. Also, this shows that the GBM can be changed from an alpha-1-2 to an alpha-3-4-5 network. Also, we basically rescued this blebbing phenotype, not totally, but inducing at 3 weeks and waiting 21 weeks, you can see how the basement membrane denuded of podocytes is now much more smooth than it would have been if it didn't have the alpha-3-4-5 network. Not as smooth as wild type, but basically these mice do live a lot longer when they have rescue beginning at 3 weeks of age. We only had one mouse die. This represents one mouse dying. But all of the mice have lived longer than their litter mates with Alport syndrome that don't have the rescue, all of which had died by 32 weeks of age, the midpoint being about 22 weeks of age. And since we published this, we had 2 mice that lived to 15 and 16 months of age, and we actually sacrificed them to see what was going on, and they still had alpha-3-4-5 in their glomerular basement membrane, but they did start to exhibit some signs of Alport syndrome. And so we've induced at these different ages. Induction at any age will lead to incorporation of the alpha-3-4-5 network into the pre-existing abnormal basement membrane. This mouse was induced at 53 days. It died just a few weeks ago at 9 months of age. And you can see that this glomerulus looks essentially normal. I'm not exactly sure why they died of kidney failure, but they did have kidney failure. They had significant proteinuria. But we're waiting, this just happened recently, so we're waiting on the electron microscopy and some other data to try to make a conclusion as to why they didn't survive. So when thinking about potential targets in Alport syndrome, there's this deranged matrix deposition, matrix degradation in the glomeruli that's associated with the splitting, including trying to target podocytes to maybe make them feel more normal. There's the significant inflammatory response in glomeruli and elsewhere in the kidney. There's, as what I showed you, there's the defective basement membrane itself. I would say that our evidence suggests that that should perhaps be a target, either by gene therapy, by infusing cells that could maybe help secrete the missing collagen, or by using a collagen replacement approach whereby the proper collagen could be infused somehow and reach the basement membrane and incorporate. So I would like to thank you for your attention, and also thanks these individuals in my lab that contributed, my collaborators at WashU who built and have been using the super resolution microscope, these investigators for antibodies and plasmids, and of course the National Institutes of Health and the Alport Syndrome Foundation for funding.